Okay, so welcome everybody to another Fungisphere talk. This talk is from Stephen Stanton, and I'm going to hand over to Philip Kwok, who will do the introductions, and he'll also lead the discussions later. So this is a talk from another one of the research themes, and I'll let Philip introduce a little bit about that theme as well. So Philip, please.
Thanks, Justin. Hello, everyone. So today's talk is under the Therapeutics and Global Health Systems theme, which I'm leading. And this theme is about dealing with fungal infections, and in particular, how we can improve the treatment and the prevention of fungal infections. So today, we're very happy to have one of our PhD students from the School of Pharmacy, Mr. Stephen Stanton. Stephen is actually a pharmacist, so he studied undergraduate pharmacy with us. And after he got registered as a pharmacist, he worked for 5 years as a pharmacist in the community. And then after that, he spent a few years in Boehringer Ingelheim, which is a multinational company with the headquarters in Germany. And he was involved with launching Spiriva in Australia. Spiriva is an inhaled medication for treating chronic obstructive pulmonary disease. And after a few years at Boehringer Ingelheim, Stephen returned to our school to study PhD. His main supervisor is Professor Di Hibbs, who is in the audience, and I'm his co-supervisor. Stephen, you also work with Paul as well, right, in your project?
Yeah.
Professor Paul Groundwater, who is here as well. And Stephen's project focuses on co-crystals, which is a way to improve the delivery of hydrophobic medications. So I'll hand over to Stephen. Thank you.
Thanks very much, Philip. I'll just quickly share my screen, guys. I've got it already. Please let me know if we can all see that nice and clear. Beautiful. Okay, so thank you very much, guys, for having me here to talk to you today. It's a real privilege to be able to talk to such a nice, interprofessional discussion with a lot of different backgrounds. I feel very honored to be able to share my studies that I'm working on, and what we're looking at is Fluconazole Co-Crystals, weak interactions providing strong results. So what we're looking at first and foremost with the world of the pharmaceutical ingredients, we're looking at from day dot in terms of discovering or attempting to discover a new medication right up to the manufacturing and commercial distribution of it. We do find that there can be quite a few instances or limitations that we do need to overcome. Now, sometimes these limitations aren't preventing something reaching the market. However, they can show a bit of an implication after it's actually being patented and commercialized out there on the market. No, nothing here is really, exempt from these kind of issues. Some tend to provide a bit more, make it a bit more apparent in the real world, others not so much. Fluconazole, I'm sure we're all very familiar with it. One of the most common antifungals out there on the market. Nice broad spectrum, great effective nature. There have been some recent resistances that have developed, Candida glabrata and Crusai, that have obviously shown a few little barriers to fluconazole's use. But what we're focusing on today and what my research tends to target is those pharmacokinetic properties. So what we're looking at here is predominantly polymorphic transformation. And I will get into that a bit more in terms of what that means and what exactly it represents. However, that does in turn lead to what we find in poor solubility, a lower stability, and then that in turn again implicates what dosage forms we can make. Now, obviously, we're looking at fluconazole or an antifungal in terms of we'd love to have that application in a lot of different varieties of infections, predominantly those who also are immunocompromised and susceptible to common things that can be quite lethal, such as respiratory infections. And if we can provide something that is able to be formulated and effective in a certain type of administration, we can really optimize that treatment regime. And as a pharmacist and with a bit of background in respiratory as well, I obviously understand the pivotal importance in terms of getting the right medication at the right dose to the right area. Okay, now there have obviously been some attempts at this. I'm not the first one to break ground, unfortunately, on fluconazole revitalization, but what I'll go over is a few other attempts that have been out there on the market, or academic circles, sorry, they're not to market, just because of some poor results that have been found, and how our attempt is overcoming those barriers and hopefully ticking those boxes. So what we'll be looking at predominantly is solvates, salts, and hydrates. So first of all, we'll start with, as I said, what is polymorphism and why is it important? And now the best way that I guess I really can explain it, I mean, it does have a definition, you know, the rearrangement of a solid material to exist in multiple crystalline forms, is a bit of a mouthful. And sometimes I like analogies in terms of getting the point across. And one of the best analogies I could think of in terms of explaining polymorphism is we go back a step. And if I said to you, you know, you've got a diamond and a lead pencil or graphite pencil, they're both made of carbon. However, they both show very, very different properties. A diamond, one of the strongest materials on Earth, whereas most of you could probably snap a lead pencil in half with your hands. How can this be, considering they're both the exact same element, but just in a different geometric formation? Now, they're what's called an allotrope because they're the same element going forward. What we're talking about in terms of a polymorph is we expand that from not just being one element to one molecule or a few molecules or a few atoms arranged into a crystalline structure. So one polymorph will be one crystalline structure, and another form of that polymorph will be just a rearrangement of that geometric formation. And obviously, as you saw from the allotrope example, you can have very different physical characteristics or physicochemical characteristics. Some of those will be, as I just mentioned, solubility, stability, and dissolution rates. So a lot of these things that are very pivotal in getting, as I said, the right amount of medication to the right site at the right time. We want it to be absorbed. We want it to get into solvent. We want it to be administered easy. We want it to be effective. Now, some examples of polymorphs that have reached a commercial stage only to trip after the finish line, you know, be disqualified or recalled in some instances, you may be well familiar with. One of the more common ones was in the late 90s, you know, peak of the HIV kind of crisis. We're talking about the antiviral medication from Abbott Laboratories, ritonavir. Okay, so that medication, its initial capsule form actually started to present polymorphic transformation from form 1, which was very effective, to form 2, which had, you know, horrible dissolution and even failed dissolution requirements, certain properties that made it very unavailable in terms of getting the right medication to the patient at the right time. Obviously, with HIV treatment, this is quite a important and lethal disease state that needs to be treated effectively. So having that polymorphic transformation was quite a bit of trouble, and it even got to the stage where Abbott had to essentially shut down their manufacture of it because the transformation in polymorphs became so rampant that the only one they could actually start to produce was the second form, which was the obviously unfortunate one that didn't work as well. So they had to go right back to the drawing board to get that reformulation redone. Another example is the anti-ulcer medication ranitidine, or Zantac, that you might be more familiar with the name. Now, Zantac was recalled recently, but not for polymorphic issues. The polymorph transformation was actually during the formulation stage where they patent one form of the polymorph, and then they started to notice throughout their formulation a second form that actually had more beneficial characteristics started to show itself. So what they realized was, well, look, we'll patent that form as well. And as the form 1 for the ranitidine started to get closer towards the expiry date, other companies got quite upset because they realized form 1's not the one that they want anymore. They wanted form 2, but that patent had been extended as they knew about it solely, and you know, they tried to go into a bit of an argument over it. But it just shows sometimes there is a benefit with polymorphs, but other times there is quite a negative. And these kind of structural differences can form quite a big impact in terms of how a medication is simply formed as a dosage form, administered, and how effective it can be in terms of a therapeutic guideline. So what we're talking about, back to fluconazole, is how polymorphic transformation of fluconazole can start to impact a lot of these physicochemical properties. And the commercial availability of fluconazole is in the anhydrous form 2, as you can see here. Unfortunately, it can be transformed to anhydrous form 1 through different conditions, but even also to a monohydrate. And that's simply the integration of water molecules throughout the crystal lattice of the fluconazole itself. So what we're looking at here is a most soluble form in the anhydrous form 2, less soluble in the form 1, and an even less soluble in the monohydrate. Now, a few papers have looked at what the causes of this, or the driving force of this polymorphic transformation is, and they found a few different conditions that can lead to it. One of the first ones, as you could imagine, being a monohydrate, is the level of hydration. So how much relative humidity in the air and at what stage does it start to impact in terms of the complete transformation from the anhydrous form, so absence of water, to being the monohydrate. Now, this can obviously impact anywhere from the production line in terms of in the factory right up to the dispensary shelf in the pharmacy. A lot of these things, you might see desiccants in your bottles or pharmaceutical medication sometimes. It's to try stave off that relative humidity, almost try to keep it at 0, just due to a susceptibility of the medication to transform. Another unfortunate restriction is pressure. So with an increase in pressure on the anhydrous form 2, they saw a readily conversion to anhydrous form 1. So what this does is it does limit the dosage forms available. Fluconazole only comes in a capsule form in terms of an oral formulation, and that is because the tableting ability, which does require quite a bit of pressure, unfortunately does then hence reduce the solubility if it does promote a formation of anhydrous form 1 from form 2. And then finally, there is also a time limit. They did find in some studies that a certain time elapse would cause this polymorphic transformation as well. Whether that just means they couldn't put their finger on what exactly it might be, or they just found that under the same conditions, anhydrous form 2 stabilized into an anhydrous form 1 polymorph. And this obviously does impact the shelf life, or how long we can keep that more effective structure of anhydrous form 2, the primary one. So all these issues or limitations that seem to impede fluconazole getting to where it really is the golden standard tends to lead to other attempts. And as I said, we did have some in academic circles that I've gone through in my studies to try to see what they've done and what limitations they might have had. And one of the first ones was a solvate that I noticed. Now, there is only one solvate that I have tried. And what a solvate is the active pharmaceutical ingredient, fluconazole in this instance, bound with a solvent molecule. So in this paper, they used ethyl acetate as the solvent, and they were able to create an ethyl acetate solvate. So a uniform integration of the fluconazole and ethyl acetate. It is in a liquid form. So in terms of dosage limitations, there is that limit there. They unfortunately didn't have any physicochemical results, so no measure of dissolution, bioavailability, anything such as that. However, ethyl acetate is a generally regarded as safe item. Now, when I say generally regarded as safe, I do mean in regards to a GRAS list. Now, the GRAS list is a list of FDA provided list of all ingredients in terms of safe for human consumption. So essentially, having it on the GRAS list, it does give it a bit of support in terms of, oh, this is a viable option for commercial application. However, there are certain limitations as to what it is approved for on the GRAS list. So whether it be food coloring, flavoring, pharmaceutical, animal only or veterinary only products, etc. We do need to look into it a bit more. However, it is promising to see that there are some GRAS list items that can start to improve the physicochemical properties of fluconazole, just maybe not in the solvate that we're looking for. Now, a second product that is pretty, unspoken was apparent is the hydrate. So obviously the monohydrate is quite readily formed in the wrong conditions. So as we saw before, the solubility is a lot less than any of the anhydrous forms. So unfortunately, the inferior physicochemical results kind of put a dead end on that one, and no one's really looked at in terms of using that as a formulation. Now, the salt formation is one of the key attempts or key aces up the sleeve in terms of the pharmaceutical industry in improving pharmaceutical ingredient properties. So salt formation might account for, say, 50% of the pharmaceutical products to date that have shown great benefits out in the community. Unfortunately, salt formation does require a few rules that don't apply to every medication or chemical entity out there on the market or at university research. Now, what a salt formation requires is an ionic center in your molecule. So it needs to have a counter ion to be able to have a positive negative attraction to accumulate into a medication. Now, there have been two examples of a salt formation for fluconazole. One was using picric acid and one was using oxalic acid. Now, because of fluconazole having a very weak basic nature, it needs a very strong acid to balance that out. And unfortunately, using such a very strong acid brings us back to a bit more of a commercial applicability, cost of manufacture and safety when using it. It might show great benefits, but in terms of a safety nature, maybe not the most ideal combination. But we're getting close. We're getting closer. Hopefully our ones, and with our results, they are looking promising, that we are finding that right balance. It's just really understanding the structural formation and being able to preempt any polymorphic change to keep these physicochemical properties that are enhanced moving forward. So we start to look at co-crystals, and co-crystals is one of the key areas that I'm looking at in my lab. And co-crystals, in compared to salt formation, is a very, very, very new technique. When I say very new, I'm talking, 20, 30 years, but in terms of the medical research industry, 20, 30 years is a very short time. So what is a co-crystal? Now, a co-crystal is a combination of two or more molecules via weak intermolecular bonding. So there's no covalent bonds. We're mainly looking at hydrogen bonds, van der Waals forces, and pi-pi stacking. Now, the benefit of having these weak intermolecular bonds is there is no functional or actual change of the active ingredient itself, no modulation of it, just simply binding of other ingredients to it that will dissociate upon administration. So dissolution will find the fluconazole and the second ingredient separate. So you're left with still the same medication, the fluconazole, just at a much more stabilized approach. Now, I put in a diagram here just to show, I guess, recap of the different forms we're looking at, try to put it, I guess, visually into a bit more perspective. The salt that we're talking about we needed that counter ion balance. So we needed the positive and the negative difference for attraction and accumulation. The solvate or the hydrate, we're looking at the stacking of the active pharmaceutical ingredient in blue. And then obviously with a water or a solvent molecule, in that case, it was the ethyl acetate. The polymorph, as I mentioned, same ingredient or same molecules, just rearranged in a different geometric formation. So no addition of anything there, just the same thing rearranged. And then finally, our co-crystal. Now you'll notice co-crystal, it looks very similar to these, but the main difference here is the neutral guest. And the benefit of that is, and one of the strong advantages it has over your salt formation or the solvates, is the co-crystal co-former or the second ingredient doesn't require that ionic center. It doesn't require a counterbalance to bind with this medication. All it really requires is a complementary binding moiety. So hydrogen bond donor or acceptor sites, pi-pi stacking regions, different regions in bonding, those weak bonds, for it to find the right parking space, so to speak. Now, what that means is because it doesn't need an ionic center, it essentially opens the door to almost any medication can be co-crystallized, as long as you find the right co-former. And because we're looking at just general structural differences in terms of binding, not so much structural and geometric, but binding differences, it has a greater potential for co-former selection as well. So not only do we have a higher starting point or a greater spectrum of a starting point medication, but also we have a greater complementarity selection as well. Now, one of the great benefits we have of co-crystals, and this has been seen from day one, is has a great potential at preventing polymorphic transformation. As you can imagine, the addition of a second ingredient or a co-former, it restricts the formation a bit in terms of how things can move around. If you're all sitting in the back seat of a car and you've got two people sitting there, you know, we can have different configurations in terms of who sits where. If you add a third person in there, there's not as much moving around and everything tends to be stuck in place a lot better. So with the co-crystallization, we found that polymorphic transformation was subdued quite significantly. And that's not endemic just to the fluconazole, it's also to other medications that have undergone co-crystallization. Now, the other benefit of co-crystallization is there are multiple methods of formulation. So what I mean is how we make the co-crystals. So we can range from laboratory bench, where we're just making a small thing for screening, which we might use a solvent evaporation. So we dissolve the active pharmaceutical ingredient and the co-former into a solvent, and we let it evaporate over a certain amount of time. Controlling the temperature might control the speed at which the evaporation is. The slower the evaporation, the more time for these molecules to align better and make a substantially larger co-crystal or actual crystal itself. Or if we allow it to have a fast evaporation, we get more of a powder residue. And this can be dialed right up to a commercial level where there's forms such as hot melt extrusion, which uses a sort of corkscrew nature where ingredients are passed into a continuous motion. Heat is applied until they melt and interact together and then come out in a cooling phase where it is a dynamic process. So these things are continually being made rather than having to start it with certain batches, which does make it a much more feasible and you know, a time-effective production, as long as obviously they are not undergoing any kind of transformation or unforeseen. But that again is what we're doing here today, looking at the structure and trying to prevent that. Some of the other regulatory bit of a paper kind of benefits of co-crystals, as you saw from that Zantac example, is you can have intellectual property extension. So you will find that some medications that are reformulated, they do start to show a different form that you can be patentable as well to extend, I guess, the life of the medication or the new form of that one. It's similar to that second polymorph being extended in regards to the first one for the Zantac. There is also the regulatory recognition of co-crystals, and the FDA in 2018 defined a co-crystal as simply just another polymorphic form of the pharmaceutical ingredient or the API, which means that retrospective clinical trial data can be applied prospectively as well to that co-crystal as well. Since there is no modulation of the actual pharmaceutical ingredient, it's still considered the same ingredient, just in a different form, which is obviously very, very, very time saving and cost effective as well. So obviously with this absolute abundance of benefits, you could imagine there are co-crystal attempts already for not just fluconazole, but a lot of medications out there. Now, considering it's been about 10 years, this is all the current fluconazole co-crystals that there are to date. Now, what you can see is, coming from a chemist perspective, I'd see it straight away and I go, oh, dicarboxylic acid, perfect. I think co-crystal, and I think, well, I need to find those hydrogen bond donors and hydrogen bond acceptors to help kind of bind it together, get it nice crystalline. And so what I see here when I see this table, I see that. Now, I understand I'm not talking to everyone who's a chemist, so I have done the nature of pointing these ones out, but you'll see the repeating organic structure here of the carboxylic acid. Some of them have two, some of them just one. And what we're looking at here is these are very, very complementarity to the structure in fluconazole to be able to bind in certain hydrogen bond accepting regions. So there is a lot of success, as you can see, 8 different co-crystals already. And we obviously took the liberty in using one of these because, you know, if it's been done, that's good because it reassures us we're not going to waste time trying to make something new, when what we're trying to explain here is why these physicochemical benefits are apparent from the actual theoretical chemistry behind it. So what we did is we picked glutaric acid. We followed on one of the methods here where we mixed fluconazole and glutaric acid in ethyl acetate. Now, we did want to get obviously the control here. We wanted to get a fluconazole anhydrous so that we can compare the structure in that one in a crystalline structure to our co-crystal and then do a bit of physical testing and theoretical testing to see what the differences were physically, and then how that correlated to any kind of theoretical changes in the chemistry as well. So what we've done here is we've dissolved the active ingredient in the solvent and gone with slow solvent evaporation. Unfortunately, as I mentioned previously, that polymorphic transformation of fluconazole is so rampant. Even when we're looking at fluconazole in a solvent with no water, we're getting the monohydrate come out. Now, this could be due to a number of things: relative humidity in the laboratory, potential of water content still in the ethyl acetate because it's not 100% dry, or even just simply a time restriction that, if we weren't quick enough to catch it as an anhydrous. So we had to work with what we had, and we had the monohydrate crystal. So we worked with that one going forward. Now, we did have some theoretical data for the anhydrous from previous studies we were able to use, but for now, we're looking at predominantly the crystal structure differences between the monohydrate and our co-crystal. We were able to get, obviously, the co-crystal with the glutaric acid as seen here. So first things first, we needed to look at the co-crystal in two forms. As I mentioned earlier, if we do a very slow solvent evaporation, what we're trying to grow is literally a crystal. Like we're talking like a few millimeters big, quite a large, it looks like a bit of glass. Yeah, so we're trying to grow a single crystal. And what we're going to do with that is we'll use techniques that are a bit niche, but it's called X-ray or single crystal X-ray diffraction. So we're going to shoot some X-rays into this crystal, and when they, as you can imagine, light going through a bit of glass, it tends to move a bit. This X-ray will get diffracted off the electrons in that crystal and will get collected in a receiver. Now that receiver will back calculate to give us an idea of where these electrons and these atoms are, and then hence give us our structure. So single crystal X-ray diffraction is going to help us give us our structure, know the arrangement of the atoms in that crystal, both the monohydrate and the co-crystal. And then we do a faster solvent evaporation so we can get that powder form. Now, what we want in that powder form is because we need the powder to be able to do the physical testing. So to do the dissolution testing or the TGA testing, which I will get onto in slides further on. But what we need to do is make sure that the powder that we've grown, first and foremost, is a co-crystal and not just simply two individual ingredients that have evaporated out. And the other way we do this is, or the predominant way we do this is powder X-ray diffraction. So on one, we have single crystal X-ray diffraction. On this one, we have powder X-ray diffraction. And as the name suggests, instead of shooting X-rays at a single crystal, we're shooting it at a powder bed. Now, when that diffraction comes out of that powder bed, it'll give you a diffraction pattern. Now, think of this diffraction pattern as a fingerprint for that molecule, okay? So this diffraction pattern is unique to the fluconazole anhydrous form. So using the powder, we were able to get that diffraction. Using the powder for the glutaric acid, we're able to get this diffraction pattern. And to differentiate between whether it's just a mixture of two individuals or it's an actual co-crystal powder, you see there should be a summation of these two if it's a mixture. Okay, so these peaks should all just be added together on one big diffraction pattern if it is a mixture. If it has a unique pattern, it means that there is a unique molecule there. And luckily enough, that's what we had here with our co-crystal pattern. So what that did is it just confirmed for us that the powder we'd made is the co-crystal, which is perfect. We can use that for our fluconazole testing going forward, the co-crystal testing, sorry. The second thing we did is we had our single crystal that we grew, and we did some single crystal X-ray diffraction. And what that happened to help give us is a structure that I'll go on to as well. And by analyzing that structure, obviously, as I mentioned before, the polymorphic transformation can influence these physicochemical properties quite significantly. A single crystal or co-crystal structure will also change the properties quite a bit. And what we need to do is we have the ability through single crystal X-ray diffraction to understand what the structure is and translate that to the physical properties. It's one of the big gaps in terms of a lot of the crystal or co-crystal studies is a lot of people are making these co-crystals and seeing the results, but not many people are understanding why that has that result. Being able to identify why you have that result, you can further extrapolate that to other medications with similar structures or similar bonding and look at applying certain patterns and enhancing them. It saves a lot of time and a lot of effort, plus it removes the need for trial and error, which is obviously quite cost demanding and time consuming. So what can we get from the structure and translate that to in terms of theoretical reasoning? What we tend to look at is three key things. We look at the empty space within that crystal structure, so the void volume. So what that empty space is, that empty space in the crystal structure gives opportunity for certain solvents or water molecules to get in and start to degrade these crystals or co-crystals. So what you mean is, if we can do a co-crystal that has less void volume or a tighter pact, we'll have less room for solvent to get in and hence a much better stability. Greater stability gives greater prevention from polymorphic transformation, but also a longer shelf life, which is great. We also look at the molecular electrostatic properties, the MEP, and what that essentially, molecular electrostatic potential, MEP, and what that looks at is the redistribution of the charge. So if we have certain positive regions of a molecule and negative regions, but we add a second ingredient, a co-former, the glutaric acid in this case, you could expect that the charge is going to be redistributed. And we'll go into this pictorial diagram as well to show the actual effects of that and what that redistribution of charge means. A positive region that used to be there might be a bit more neutral now. So in terms of when we layer these molecules on top of each other, obviously positive and negative would have that attraction together. Similar in the vice versa, a negative would have a repulsion. And that kind of dictates the geometric formation. So if we start to form more neutral zones, we start to find they can pack a lot closer because the repulsion is removed. Now, the final thing we're looking at here is the lattice energy. So what holds it all together? All the summation of the intermolecular and intramolecular bonding. So those weak interactions, the hydrogen bonds, the pi-pi stacking and the van der Waals, how do they all summate to keep it a more stable molecule, but also to look at the entropy impacts as well. Now, the entropy is the measure of disorder, and we'll go into that one a bit more as well, but these are the theoretical chemistry applications that we're looking at in terms of explaining or correlating, the physicochemical properties. So first things first, I know when I started talking about single crystal X-rays, you guys were like, oh, he's going to show us the structure, and here it is for you, okay? Here's the monohydrate that we're looking at, and here is the glutaric acid co-crystal with fluconazole. Now, I know this, for me, as a chemist, I see this and I go, oh, perfect. You know, it's a finder word. I can see everything right here, and I know what I'm looking for. For biologists, doctors, other people, it takes a bit of time, but what I've gone here is to show you where exactly the key things are looking for. And as you'll see, these are the fluconazole molecules, and these are our water molecules in the monohydrate. Now, as you can see, all these water molecules are aligned here in a hydrophilic column. Okay, so all the water loves to hang out here. And what that does is that starts to orientate all the fluconazole molecules to push all their hydrophobic ends out here. All these fluorines start to stack on opposite ends here. So all the attraction and the hydrogen bonding here starts to rotate it and push it the other way. When we look at the glutaric acid co-crystal, when we start to look at the glutaric acid co-crystal, we don't notice these straight lines in terms of sheets. We notice more of a zigzag, okay? So these zigzags of fluconazole molecules and glutaric acids start to give a bit of a tighter squeeze, with everything start to groove or fit into grooves a bit easier, which obviously, as I mentioned before, when we're talking about void volume, obviously the more snug they fit, the less potential for these solvents or waters to get in, which means a greater stability. So when we look at that hydrophobic region here, that's probably where a lot of these solvents will start to penetrate, because there's a lot of empty space here as there's no attraction between them. Similar thing again, we do see it in the glutaric acid co-crystal, especially between these pi stacks right here. And what we had then to do is use some theoretical software to try calculate how much space is in there and whether there is much of a difference. Another view of this 3D co-crystal, just to try to give you a better idea of where these columns are, we start to see right down a different angle that these are more like tunnels right through the whole crystal structure, which means that water is going to come right through here, but it's not going to separate out the molecules. So we probably start to hypothesize that the dissolution of this one is not going to be as good as something like the glutaric acid, which essentially means when you go to take this tablet or capsule in your stomach, it will take a while for it to kind of break up and dissolve into the, or spread out into the stomach acid or the solvent or glass of water if you're trying to dissolve an electrolyte tablet, effervescent, sorry, tablet in there. So having these water channels will mean a lot of water isn't really going to penetrate to separate out these molecules, and it will be a bit slower to dissolve. So we talked about the void volume, and from our calculations using software, we found that the fluconazole monohydrate had about a 10% void volume. That means about 10% of that crystal structure was just empty space, 10% room to rent for these solvent or water molecules to get in and amongst it. And you'll notice the regions that they are appearing is obviously in that hydrophobic channel there. Okay, when we look at the fluconazole glutaric acid co-crystal, yes, again, similar hydrophobic regions, but only at 4%. So that shows there's a much tighter, much more snug fit, which means a much more stable molecule going forward, which is a great theoretical supporting case for hopefully some good physicochemical results. So we talked about the MEP, molecular electrostatic potential. And what we were talking about is the positive and the negative regions and the difference in charge across that molecule. Using the legend here, you'll see purple and red zones, which are quite opposite ends of that charge spectrum. The bluish purple are hydrogen bond acceptors, whereas the red ones are hydrogen bond donors. So usually on the end of these water molecules, on the hydroxyl groups, and the other ones, the acceptors on the nitrogens. So you see quite a strong variation in the monohydrate form. When we look at the glutaric acid co-crystal, a simple addition of this co-former, this glutaric acid, look what it does to the fluconazole charge. It obviously reduces a lot of these strong red and blue areas to more of a neutral yellow and green. And as I said before, what that tends to do is having these neutral zones means we can stack them a lot closer. There's not as much repulsion between these charges, which means the geometric formation means it's going to be a lot more stable, hence warding off any polymorphic transformation, which is, again, something that's very, very positive. So which one is held together stronger? So in terms of the lattice energy, we're looking at a few different values, namely a melting range. So the higher the melting point or the melting range, we're looking at the stronger these bonds are held together. So it's the level of energy needed to overcome to break these bonds and melt it from a solid into a liquid form, etc. As you can see, the fluconazole glutaric acid co-crystal had a much lower melting range than not just the monohydrate, but also the anhydrous form as well. As I mentioned, we did take this data from previous studies, but we were very happy when we saw that ours was a lot better, which means a much higher solubility. So if it has weaker bonds in there to break apart, once it's dissolved, that means it's going to have a higher solubility in terms of getting absorbed into dissolution. Now, another measure is the entropy. So another method for predicting the solubility of a medication is the solid state entropy. Now, entropy is a measure of disorder. I guess the best analogy is if you were walking, a class full of students through a busy area full of a lot of people. If you had them in a very structured, rigid state trying to get through, it takes a lot of time, a lot of effort, a lot of force to try to keep them in that state, pushing through. If they were a bit more looser, a bit more freedom to move, flexibility, it makes a lot less potential needed to get them through the swarm and out the other end. And what we're talking when we translate this to a physicochemical state is a higher freedom of energy or freedom of movement in terms of entropy for these molecules or atoms in the molecule means a higher potential or a faster potential for them to break free and get dissolved in amongst your solvent or your glass of water or your stomach, wherever you're administering this dose. So we're very happy to see a higher level of entropy in terms of our co-crystal compared to the monohydrate and the anhydrous form. So this is a lot of theoretical results here, guys. This is a lot of strong benefits, potential hypotheses that we needed to really make sure correlate with our physicochemical results. So we bring in our powder again, our powder co-crystal, and we start to do some physical dissolution testing. So dissolution testing is essentially packing a certain quantity of the powder into a flow-through cell. And what we do is we run a certain liquid through there. We mimic the gastrointestinal fluid in this instance, so temperature, acidity, etc. And we take samples at different time points, and we measure how much fluconazole was in that sample at that time, okay? And as you can see, the results straight off the bat, at time point of 2 minutes, we had about almost 70% of our fluconazole dissolved into solution at 2 minutes. When we compare that to the monohydrate form, at 2 minutes, we had just over 10%. That's quite a considerable difference in terms of speeding up that dissolution, which means we can get more medication into the body at a faster rate, which is a very, very promising result. When we start to look at the 10-minute mark, we notice our co-crystal is almost at 90%, whereas the 10-minute mark here, we've only just reached 70%. For the monohydrate to reach 90%, it took about 20 minutes, whereas our co-crystal took only 10. So doubling the speed of dissolution was an absolute, very, very good news to reaffirm a lot of the theoretical studies we've done based on the structure. It just reaffirmed that potential for those hydrophobic regions, or the better stacking, for it to dissolve and break free a lot quicker than simply allowing water molecules to get through those hydrophilic channels that we saw in the monohydrate. Now, when we look at the stability of our molecule, we're looking at how much water it can absorb into its structure. So this doesn't just generally mean bonding into the structure, but it physically means absorbing a water molecule from the air into the actual physical product, whether it be a capsule or a tablet, which can unfortunately cause degradation and a lower shelf life. Now, what we did here was called TGA, a thermogravimetric analysis. And what it is we stored both our co-crystal powder and the monohydrate powder in different relative humidity chambers, ranging from 84% down to 0%, stored it for a week, and then we put it in the machine that will heat it up from about 25 degrees up to about 130 degrees. And it measures the mass change as it heats it up. Now, obviously, as it heats up, water is going to evaporate off, so the mass is going to get lower, okay? The greater the amount of water that has been absorbed in that week storage, obviously the greater mass change you're going to have. Now, I realize that these colors are a bit overlapped here, but I can safely say and happily say that all these colors here were our co-crystal samples, showing almost 0% weight change or mass change. All of these other ones were the monohydrate at differing levels of relative humidity. Obviously, they've absorbed different amounts. What does this mean in terms of application? This means that our co-crystal, reaffirming our theoretical results, really is going to be a much more stable product in terms of relative humidity. So we looked at that humidity transformation from the anhydrous form 2 to the monohydrate. This one's going to keep the co-crystal for a much longer period at almost 84% relative humidity going forward, which is obviously a quite considerable amount. But again, we can have high amounts in the manufacturing process along the factory line. But this is a great application in terms of providing that stability and structure going forward to slow down any degradation, but also polymorphic transformation that can cause a bit of trouble. So that's today's situation. That's where we've got to so far in my research. What I'm looking forward to next is really starting to get into what tomorrow's formulation could be from all this great promising results. Now, because we did formulate, I formulated this co-crystal powder in terms of a solvent evaporation. Unfortunately, very good at finding a screening product, but in terms of physically modifying something like this, so talking about particle size control, it's not the most delicate process. We can't really control how big these powder fragments are going to be. Now, for lung deposition, as we're talking about patients who are immunocompromised, we wanted to get it down as deep as possible into the bronchioles as we can. Now, 10 micrometers is the ideal size, or around that size, because we do need to balance the turbulent air of the lungs, but also size in terms of getting into those alveoli. So 10 micrometers is what we're aiming for. In terms of physically refining the powder we already have, it is a bit of a limitation because applying pressure or any kind of energy into the system as a co-crystal may either cause dissociation or even a polymorphic transformation because we're applying energy into there. So ball and mill grinding isn't going to be an option for us to go forward with that one. What that does need for us to look at then is other co-crystallization methods. As I mentioned before, there's quite a few that can be used to control the physical size of the product. Now, there are a few formulations that have become, or methods that have become a bit more apparent in the last few years, so still quite fresh. And I am still trying to, browse through my network contacts at Sydney Uni and even private institutions to see any availability of these resources. But one of the first ones is a supercritical anti-solvent. So dissolving all your ingredients, your API and your co-former in a large solvent, and then pumping through a second solvent that would provide a different solubility for these products. So as the solubility changes or the concentration changes, they start to precipitate out in a very fine powder form that can be controlled based on the solubility difference or the speed. The second option is spray drying. Now, spray drying is one of the more common options for aerosol formation. However, there is the risk, because it is still a very new nature with co-crystallization, there is a risk of possible dissociation upon spray drying. So you still might not get the co-crystal itself. You might get two individual ingredients that are dissociated. It is an option to try, but again, I'm still trying to cover all bases going forward there. And then finally, of course, hopefully when I get to it, we can start to do some cytotoxic assays as well, basing the co-crystal fluconazole against the anhydrous form in terms of finding out the AUC, the cytotoxic events, and how much we need to really get the same therapeutic window, and looking at side effect profiles for a co-crystal compared to individual. Yeah, so as you can see, quite a big project here, and I'm being very lucky to have a lot of help and support along the way, both in terms of guidance and also, just simply how to do things. I'd like to thank my primary supervisor, Professor David Hibbs, and Paul Groundwater. My apologies for not having your name there, Paul. I'd also like to thank Dr. Jonathan Du, Dr. Sam Duca, Dr. Philip Kwok, Lace, Bryson, Felsia, Penny, and Wai Ting. Helped out with a lot of different steps in different formulation testing methods and theoretical calculations. I'd also like to thank the Australian Government Research Training Program Scholarship and the Fungisphere seeding funding for making such an exciting project possible. Thank you.
Thank you very much, Stephen, for the interesting talk. So now we are open for questions. Does anyone have any questions for Stephen? You might like to ask verbally, or you could type your question in the chat box.
Oh, Philip, I can ask a question if
okay,
it's okay.
Yes.
Yes, I was just wondering why. I mean, it's news to me, I guess, that fluconazole has problems with solubility because it's used as a capsule, to treat thrush, and it's taken orally. So so firstly, I guess that's just a comment. I didn't realise that there was, when I think of solubility issues with antifungals, amphotericin B usually comes to mind. And I had a bit of a chat with Justin, and he reminded me that that's administered intravenously, not taken as a capsule. But yeah, I guess it just, that did surprise me. And I guess what I wanted to ask you is that if there is a problem with solubility in a capsule form and you would like to consider administering the drugs as a co-crystal. I just remember from some of the work, the collaborative work I did with you, Philip, where we used a different co-crystallizing agent, that there was quite a high proportion of that reagent in the final complex. And so it drastically increased the molecular weight of the final product. And so I just wondered, so although it's very clear that the solubility is enhanced twofold, is the fact that you have a much bigger drug now going to compromise how big a tablet can be when someone takes it? So you're sort of trading off solubility for taking a tablet that's really huge.
Yeah, great question, Julie. So in terms of the effectiveness of something like this, if we're able to find that we can get a faster rate of solubility or a greater distribution of a dissolution in terms of the active ingredient in there, we might find that actually the quantity needed of the active pharmaceutical might actually be lower than what was needed originally when it was by itself to get the same therapeutic effect. So you might even find there is a reduction in certain primary active pharmaceutical in combination with a co-former. The bioavailability of fluconazole is quite high, so there's no limitation there. But in terms of how much is dissolved to be absorbed into distribution for bioavailability, you know, if we can increase the amount there, you'll find that you won't need as much. So obviously fluconazole being the much larger of the two molecules, you know, reducing some fluconazole for some glutaric acid, you probably have a net still positive overall going forward in terms of molecular weight. We might also find that the certain dosage range might not be needed as high. You know, it might not be the 200 to 400 milligrams. It might be a 100 to 200. Who knows? Like, as in, there is obviously going to be variations in that, and we obviously do try to find that in terms of maybe the next stage as to what kind of AUC we're working with in cytotoxic therapeutic windows.
Okay. Oh, thank you very much. And by the way, it was a really clear presentation. Very, very, very nice.
Thank you. Hopefully I didn't speak too quick.
No, it was great. I mean, if you don't mind, can I just ask you another?
Of course.
Another question. I was just wondering, we've got a collaboration with the Department of Pharmacy in North Carolina, and they're sending us compounds which are yet to be patented. So unfortunately, we don't know the structure. Yep. And they're very potent in vitro against our target protein, much more potent than anything we've seen before. And this is something that actually Des Marini, who's in the audience at the moment, is doing as part of her industry-based PhD.
Right.
And so I guess I was going to ask, in the absence of knowledge of the structure, so we're seeing a little bit of a synergistic effect of this compound with amphotericin B in a susceptibility assay. Yep. But it's not, we still don't know whether there's solubility is an issue or whether it's crossing into the cells very well. We know that, you know, once it gets into the cell, it's probably going to be very potent against its target, but it's just getting it there. So is it worth sort of trying the glutaric acid and some of these other molecules that you're talking about today in the absence of sequence information and trying structural information, and trying to, you know, use some of your, I think, NMR technology to sort of look at whether or not some co-crystals are forming and use them in antifungal susceptibility assays? Yeah,
I mean, straight off the bat, when you're talking about structure, so obviously, hopefully you guys got from my speech, connecting a lot of the structure to these properties is sometimes the absolute linchpin here. Like, in terms of finding out what the modulation or what the binding is from that structure, or the redistribution of charge, can help to sometimes explain, or even give you a bit of guidance to go, oh, like, you know, maybe if we do something here, it can help to go there. I definitely would suspect that if it's a solubility or a stability kind of issue, co-crystallization does provide an opportunity. In terms of whether the dicarboxylic acids, such as glutaric, are optimal, it does depend on the actual product itself and the binding motif. So hydrogen bond donors, acceptors, what we're looking at. If there is any samples of it, we could probably do try to do a structure determination for you, if that helps.
Sorry, Stephen, I didn't mean to say that. It's just that it's under patent. So
okay, no worries. That's fine. Yeah, no, but then, I mean, I guess guidance for them going forward, that if they were to look at that structure, it gives a bit more time-saving effort to know what you're working with. But I could definitely say that based on both my results in this present study and other ones, you're finding quite a few enhancements in terms of these effects. And obviously, as you said, getting it to the right amount to the right spot, once it's in there, it does its job. So that's half the battle.
Yeah. Okay. So do you think it's worth us waiting till they file the patent so that we know what the sequence is? or should we have a go at trying to make them more antifungal and then give them that information and say, look, you know, because they're developing these compounds for human treatment, not for like we've signed a confidentiality agreement with them.
I mean,
antifungals.
I mean, it's up to you, obviously, in the collaboration with North Carolina, the cost of manufacturing these things, whether there's enough to kind of, you know, trial and error a bit. Again, that's what it would be. In the meantime, if you have a general idea of the structure in terms of what it is, but not the exact structure, you can probably cut a bit of time down or cut a few, eliminate out a few other options and probably narrow down your search. But in the essence, it will still be a bit trial and error at the end of the day. It just comes down to whether they have enough and they're willing to sacrifice enough to throw into there. I usually, you know, I'm a co-crystallizer, crystallography, so I would say yes, but again, that's biased, unfortunately. But yeah, there's definitely merit to it if that's what you're looking for. It just depends on whether it outweighs the cost of the actual active pharmaceutical ingredient.
Yeah, okay, thank you, Stephen. Thanks very much.
Thanks very much, Julie, for the great discussion. Any other questions? I'm mindful of the time. Just one quick, yeah, it's 5:02. Just one short question from me. So during the earlier part of your presentation, you told us that the anhydrous forms were more soluble and the solvate, the monohydrate, right? Yeah, the monohydrate.
Yep.
So it's just a bit counterintuitive because you think that there is already water in the crystal lattice. If you put that monohydrate into water to get it dissolved, then it should help, right? But it actually doesn't. It actually made it worse. So
right. Yep.
Yeah, it's more of a comment. Like you told us that it's due to the way that the water is aligned, right? So
again, potential reasons can be a number of things in terms of inhibiting this one. So a more rigid structure that has those hydrophilic columns in there where water isn't really going to get in amongst in terms of separating out the other ones can start to stabilize it a bit against, which slows down that dissolution, which then inhibits the solubility. Because obviously before we are soluble in the solvent, we need to break apart the actual physical capsule itself or binding, so the dissolution comes first. So it can be a number of factors in terms of the total presence going forward before it is soluble, but it was difficult to correlate exactly the mechanism as to why, but the theoretical numbers suggested that one based on the entropy and a combination of other dipole moments and things like that, that we found the co-crystal just to be more beneficial than the monohydrate.
Yeah, certainly.
Yeah. It's good news for our numbers.
Yeah, that's great. Yeah. Thank you.
No, thank you.
So Justin, do you have anything to add?
No, just really to say thank you very much, Stephen. I thought that was a great talk. It's absolutely not my area, and it's a long time since I'm looking at chemical structures, but I thought you did a really nice job of landing the message. So well done, you.
Thank you.
Even for a simple mind like mine. Appreciate the discussion. Yeah, and I'm sure there's opportunities to work together on different things in the future for people who are in the audience.
Happy to help.
Okay, amazing. Well then, thank you, and we will end the meeting and look forwards to the future.
Thanks very much for having us, guys.
Thanks, Stephen. Thanks Justin. Thanks, everyone.
Thank you.
Thank you, Justin.
Thanks, Julie.
Bye-bye.
Bye.